Introductory Neuroscience and Neuroinstrumentation, BCI applications. Okay, so in this session we shall consider the different brain-computer interfaces, applications in a little more detail and look at trade-offs between usability, training and bitrate. So just to reiterate, so we are only considering scalp-recorded BCI control signals. And the first one is the SSVEP, and the number of choices we have are very high. There is no training involved and you have high information transfer rate. Slow voltage potentials usually seen in, usually done in patients with quadriplegia who are really quadriparesis and all four limbs are paralyzed. So here it's difficult. There's a lot of training is required and the information transfer is very low. Then the P300, the number of choices are high and there's not much training involved and the information transfer rate is moderate. And finally sensorimotor rhythms, which are where you image visual imagery of do imagery of motor movements like moving your hand, moving your leg, and here the number of choices are low but you need a lot of training and the information transfer rate is not brilliant. So let's consider the P300 BCI application speller in detail. So to reiterate, the P300 is a positivity occurring at 300 milliseconds after the brain senses a novel stimulus. For example, you're walking and you suddenly see a snake, you jump, that's a P300. Later it may be a rope or something but you still react very fast. So that's a P300. In the lab you have an oddball stimulus. You have subjects who are presented stimuli of two different categories, a rare category and a common category. The rare category, the rare stimuli elicit a P300. So this was one of the early papers, Emmanuel Donchin, in psychophysiology. It's a surprise, surprise. So this is one of the early descriptions of the P300. So coming to the right panel, the P300 brain computer interface, on the right you have rows and columns of a letter matrix which are randomly illuminated either horizontally or vertically. So suppose you want to say please. So the first letter is P. So the subject focuses on when the P comes and after 6 or so P's he will trigger the letter P. As I said earlier, this is very painful and slow for us. We speak so fast and we transmit so much information, but for a person who has a stroke or has some severe cerebral dysfunction, this makes all the difference. Okay. On the left panel over here you actually see the P300 potentials to the desired letter in red compared to the non-desired letters in green. The non-desired letters will just be the standard visual evoked potential. This is the P300. So laboriously you do P, then you do L, then you do E, then you A, and then you do S, and then you E, and you get please. Okay. So the logic is that this was using the GTEC system. So the stimuli are given, the subject sees them, this random flashing of rows and columns of different letters. Then it's amplified and suitable signal conditioning is done with GTEC systems and signal processing is done removing the artifacts, epoching, filtering, so on and so forth. But then you have a classifier, okay, and the classifier tells you whether which letter has been chosen, and either you can write the classifier yourself or there are open source classifiers. So you can do this very inexpensively by instead of GTEC you can use OpenBCI. GTEC can be afforded by research labs and hospitals, but individuals can use OpenBCI, use the OpenBCI software, and then you use an open source classifier, NeuroRemove, etc. There are many of them. Integrate all of them together and you have your own P300 BCI speller. And if you're an Indian, you could use Indian languages, Hindi or Tamil or Kannada, for your speller instead of English. The thing about BCI spellers are that right now the information transfer rate is very slow. So One of the things which could be useful would be some kind of heuristic word completion, like what you have in cell phones. You're typing and then it completes the word. So that would be really useful to increase the information transfer rate. The other thing is to decrease error rates. You use hyperscanning. So simultaneous multi-subject recording of different subjects is called hyperscanning, and researchers have done this by using either fMRI or EEG devices, which are ideal. fMRI is impractical really. And finally, you can decrease the number of trials by extracting single trial P300 events using wavelet analysis. So wavelet analysis allows you to denoise the data so that you can see single trials, P300 in single trials, as we mentioned in a previous lecture. So instead of waiting for 6 times, maybe 2 would be enough. So you'd please would be much faster. Your it'll be 70% faster than using it with 6. So coming to the steady state visual evoked potential. So we introduced it in the previous lecture. So here on the left on top is your forcing stimulus. So you have a visual stimulus over here at 11 Hertz, and below is the SSVEP, which is also following it at around 11 Hertz. And if you do the spectral power spectrum of this stimulus, you have a peak at 11, and if you do a power spectrum of the response, you have a peak at 11. So we can use this peak for the SSVEP signal. The advantage is that there is no subject training involved. They straight away, you just look at the LED, your occipital cortex is forced to at this frequency, and that allows the signal conditioning circuit to do its thing. So there's a high data transfer rate compared to the other BCI signals, and it's much more stable than just the single visual evoked potential. Okay, and this is a recording of a SSVEP, and here you see the heat map or the color interpolations on the brain, okay, of the EEG signal. So coming to the robot, okay, so this is an actual experiment where the subject is holding the SSVEP stimulus light box, and some of this work was done in the Department of Applied Physics and Instrumentation at Indian Institute of Science in Professor Sanjeev Sambandhan's lab, where the top LED blinks at 10 Hertz for upward movement, the right bottom and the left LEDs blink at 11, 12 and 13 Hertz respectively. And here you see the stimulation at different frequencies on the left, and you see the peaks, the appropriate peaks occurring after FFT analysis, real-time analysis happening on the right, and these can be used as signals. So you can actually you can barely see the robot here unfortunately, but you can use this to make the robot move in whichever direction you want. Okay. So what are the best frequencies ranges for the SSVEP robot? So you can go from 5 to 60, but and you can see that at 15 it's good, at 25 it's good. So you can choose the appropriate frequencies and for the signal conditioning circuit. Over 60 it's not really helpful, and less than 5 it can be not so helpful. Okay. So this is the average SSVEP amplitude as a function of stimulus frequency, amplitude and frequency. So coming to mu rhythms. So in awake people, the primary sensorimotor cortical areas, that's over here, they show an idling kind of an activity, like a car or a scooter in neutral, between 8 to 12 Hertz, when not engaged in processing sensory input or producing motor output, when you're just still. So this idling activity is produced by the midbrain structure, the thalamus and thalamocortical circuits. The thalamus, which is in the center of the brain, and it has recurrent circuits with the cortex, and this is the idling frequency, if you will, of a thalamocortical circuit. The thalamus is very important in both sensory, it's a relay station for sensory input for all the modalities, visual, auditory, somatosensory, etc., except olfactory. Olfactory doesn't go through the thalamus, and It also controls the motor activity. It's part of the motor circuit. And most of the thalamocortical circuit, 90% of it is controlled by the cortex on the thalamus. Okay? So these mu rhythms wax and wane. They come up and go down, come up and go down with actual movements or imagination of movement. And a linear equation translates the mu rhythm amplitude from one or several scalp locations into cursor movements at 10 times a second, 10 hertz. Okay? So when you have this mu rhythm, so when you think of moving, it desynchronizes. So it becomes kind of flat, low amplitude, high frequency, goes into beta. The mu rhythm is in the alpha range. So this is called event-related desynchronization, and its converse is event-related synchronization. We went through this graph previously, but I shall go through it again. The dotted line is the alpha range, which shows the mu rhythm the best. The dashed line is the beta range, where it shows the mu rhythm, but it's not as good as the alpha range. And the gamma range, which we really haven't discussed, is high frequencies, 36 to 40 hertz, and this peaks before your actual movement. We know the movement occurs here at this vertical line because we are also recording the muscle activity, EMG activity. Okay? So what is the protocol of the mu rhythm? So initially, users employ some form of motor imagery, like imagining the hand moving or imagining the body moving, etc., to control the cursor. But over a period of 2 to 3 weeks, and these are 40-minute to 1-hour sessions with 2 or 3 sessions per week, they learn to control the cursor. Okay, and they can't say how they do it, but they learn to control the cursor. And with imagery, they report that when they move the cursor, it's just like as if they're actually moving it. There seems to be no difference in imagining the movement and making the computer move the cursor or them actually moving the cursor. So the amplitude and also the variance of the mu rhythm amplitude, as well as the time it takes, it depends on the individual level of EEG control. It also depends on the pathology in his brain. If he's got stroke and the stroke affects the left side, then the mu rhythm would be diminished over here. That is the control of the right side of the body, but it will be normal or fairly normal on the right side. Okay. So how do you control it? So here the thing is, you have to move the cursor so that it hits the target over here, and this is the raw EEG. It's showing the mu rhythm, and this is the desynchronized EEG, when you're actually moving or imagine moving the hand, and that is a signal for this guy to move up or down. Okay. And these are the areas of the cortex. The yellow band is the sensory area, then there's a central sulcus, and the pinkish band is the motor area. So the mu rhythm arises from this area and what we sense in the EEG. So that's why it's the central C3, C4 electrodes. And the imagery of the movement desynchronizes the mu rhythm. Okay? And the feedback is the moving cursor over here. It's red rectangles. So you have to move it so that it hits this target. And this takes about 3 to 4 weeks. And as I said, you need to do this for 40 to 60 minutes, 2 or 3 sessions a week, and then you figure out how to do it. Okay? This is a very primitive way of doing it, moving a cursor towards a band. A better way to do this would be to capture what happens in a first-person shooter game, like the old games, Doom, Quake, and stuff. And here the subjects control the movement of the scenery by producing low mu, and they go left, or they go right by producing high mu. So again, it's difficult for them to explain how exactly they do it, but they learn to control mu within 3 to 10 hours. That's it. No weeks, but hours. And the consensus is the training is faster because the environment is stimulus rich, realistic, and it's motivating. You're moving, you're shooting, you're killing demons. It's better than this. This is, it's so bland compared to this. So future, stimuli paradigms would probably use, a stimuli like these, where, it's realistic and you'll train much faster. A much of this is in its infancy, so there is place for students like you to design new experiments, because all these first person shooter games, many of them, the original Doom and Quake, they're all open source. So all you have to do is use it, get the triggers out, use OpenBCI, and then you're in business. I mean, you have to do a little bit of work, but it's not impossible for an individual who is really interested to do it at home. Okay, and this is a good paper to start off with, Pineda et al. From 2003. Then slow cortical potentials. Now this was actually the first potentials used for BCI by the famous Niels Birbaumer from Max Planck Institute in Tübingen, one of the greats of neurophysiology, cognitive neurophysiology. And they used paralyzed subjects who were quadriplegic, who could not move any of their limbs. And again, it's very painful and slow. You see these potentials on top, and he is looking at different alphabets over here, and when he gets the alphabet he wants, there is a slow potential, but it's very painful and slow to do this compared to even the SSEP and the P300. But again, in a person who's completely paralyzed, this is a godsend. Without this, he would not be able to communicate at all. So even though it's painfully slow, it's better than nothing. And of course, we have to talk about the Japanese. They use the fMRI signal. Now, fMRI looks at the increase in the blood oxygenation level dependent signal, or the BOLD signal. And the argument is, or the logic is, that areas of the brain which are active will have increased blood flow, okay, which you see over here. And this is activation and deactivation, and this is on the surface, while here you see activation and deactivation in the basal ganglia and the premotor cortex. Okay, so this is obviously not practical unless you have an fMRI instrument, which costs anyway $10 million. So if you want to do these kinds of things, you have to be attached to an institute which has this equipment. Okay, so what are the best signals to use for BCI? Okay, so there are these trade-offs, training time versus bitrate with mu, P300, and SSVEP. So if you look at the mu rhythm, that is the event-related synchronization and desynchronization, the training time takes hours, okay, and the communication bitrate is not brilliant, it's pretty slow. P300 is much faster because you just have to sense what's coming, okay, and we assume that everyone knows the alphabet, so it's in minutes rather than hours. However, the bitrate again is not brilliant. Okay. SSVEP, it's as fast or faster than P300, and the communication rate is much higher. It's 60 bits per minute. So depending on your experiment, depending on the subject, depending on the cerebral dysfunction, you have a choice. You can use either the mu rhythm, you can use either the P300, or you can use the SSVEP. It also depends on what you want to do. With the P300 and ERDS, you can train the person for the whole alphabet. With SSVEP, you just have 4 or 5 signals. I mean, there's a limit to the number of LEDs you have, and you can so 4 seems to be optimal. You can go up to 6, but that's it. Okay. So I have to mention this, that a lot of the BCI research has been stimulated by defense interest. So you have electrodes now, dry electrodes. You can see on the left over here, these have multiple prongs like a comb, and even if one of them touches the scalp, that's fine for getting a good signal. And also you have electrodes from a company called Cognio, which is DARPA funded, and those fit so snugly and tightly that you can do somersaults and you still get a terrific signal without any interference. Also algorithms. We need really good algorithms so that we can get single events if required. So one of the interests in BCI is to see if there are any anomalies in the visual environment. For example, intruders, what's happening to our borders in Gualan. if there are any intruders. And for that, you need to have single event, pickup. You need to, because the intruder is not going to jump across 100 times for your P300 to be average. So here you use wavelets. Wavelets allow you to denoise the brain potentials. It also allows you to pick up individual events, okay, which is what you need. Okay. So thank you very much. BCI is a growing field, and it's one of the fields which most students are interested in jumping into, because there's something fascinating about being able to control your environment just by thinking about it. And the story is not over, it's just begun with BCI. There's a lot of place for newcomers, people who think differently, both medical students, neurophysiologists, as well as instrumentation people to contribute to the field. All the very best.